Okay, so to finish off this video, I would like to walk you through the week 13 lab assignment. So these are exercises that are very similar to the ones you will have to do on your homework. So hopefully if you watch me do this, you should be more able to do your homework this week. So question one, an experiment has a single factor, so one way, with three groups and seven values in each group. So first thing, I already think to myself, okay, there are seven values in each group times three, that's 21 values total. So total n is 21. I'll remember that for later. So A, how many degrees of freedom are there in determining among group variation? Among group, remember, is the variation between the groups. So there are as many degrees of freedom as groups minus one. So this is three groups minus one equals two. How many degrees of freedom are there in determining within group variation? That one's a little trickier. Remember, within group variation is the n minus the c. So n, the total number of observations, minus c, the total number of groups. We just said that we had seven values in each of three groups, so 21 total. So n is 21 minus the number of groups, that's 3. So 21 minus 3, 18. Finally, how many degrees of freedom are there in determining total variation? Well, that's n minus 1. I just said n was 21, so 20. All right, another one-way ANOVA question, an experiment with a single factor with five groups and six values in each group. So again, in my head, what do I think? Five groups, that's C equals 5, six values in each group, so totally 5 times 6 equals 30 total observations, n equals 30. Now we get more information. Among group variation has 4 degrees of freedom. Well, I already knew that because you told me I had five groups, so I already knew there was 4 degrees of freedom. Within group variation, there are 25 degrees of freedom. Again, I already knew that because I just computed in my head that there were 30 observations, 30 minus 5 n minus c, that's 25. So this is redundant information. You could have thought of that already by yourself, but at least it confirms what you know. And in determining total variation, there are 29 degrees of freedom. Again, we knew that, n minus 1. So clearly that question is not going to be about the degrees of freedom. Instead, the question is the following. If SSA is 80 and SST is 180, what is SSW? All right, so that's when you pull up your handy slides. This table I said you would use in lots of your one-way ANOVA questions. So if SSA is 80 and SST is 180, what is SSW? Well, that one is really easy. We know that SST is equal to SSW plus SSA. And with numbers as easy as this, we know that SSW is just 180 minus 80, that's 100. What is MSA? Well, we know that MSA is just SSA divided by the among degrees of freedom, and they already told us that was 4, so 80 divided by 4, that's 20. Okay, next one, MSW. Well, it's SSW divided by 25, the within group variation degrees of freedom. So 100 divided by 25, that's 4. Okay. And finally, what is the f-stat? Well, it's 20 divided by 4, and that's 5. Before we move to the next question, I want to show you a little trick. Could have gotten to all these answers really quickly if you just made this little table for yourself inside of Excel. How would you have done that? Well, put a row for among, put a row for within, put a row for total, and fill in the values 4, 25, and 29. Then it told us SSA and SST. So now we've put in all the information that we've received from the question. What else do we add? We know this is 100, and then we know that this is this divided by that. So those are the MSA and MSW, and then you can divide this by that, and you get your F stat. The reason this is really helpful is because the questions are not always going to have round numbers like this. It's not always going to be 180 minus 80, which is very easy to do in your head. But if you need to compute things, make this little table and everything goes easily. The other thing is, again, just like with your templates, if you have this handy somewhere on the back of your computer, you can easily answer all these questions. You can change the numbers according to the question. If they had told you that this was 8, well, there you go, you have a different answer. So I encourage you to make a little table like this for yourself that can help you answer quickly. Okay, question 3. It starts all the same. Single factor, 5 groups, 6 values in each group. It's even the same values. By chance, we got the same ones. 4 degrees of freedom in the among group variation, 25 degrees of freedom in the within group variation, 29 degrees of freedom in the total variation. This looks like it's the same question, but it's actually a different question. They give you the SSA, SSW, SST, MSA, MSW, and FSTAT. So the first thing they want is just for us to fill in the table, know which number goes where. How do we get the upper tail critical value from the F distribution with 5% significance? Well, what do we need? We need the degrees of freedom. Degrees of freedom of the numerator are 4. Degrees of freedom of the denominator are 25. And so we need to find the right F table. First of all, we check that this is the 0.05 upper tail. This is correct. Be careful because sometimes they want you to use 0.005 in the upper tail. So don't rush when you pick the table. But here this one was correct. We have 4 degrees of freedom and we want 25. So this is 1, 2, 3, 4. So it's 2.76. As proof of what I said earlier, let me just show you that this works in Excel. So we want an f.inv.rt, 4 degrees of freedom and 25 degrees of freedom for 0.05 level of significance. Let's just write this in, f.inv.rt, be careful, not dist, but inv, 0.05 for 25, and that is correct. 2.76, same as what we found in the table. Now, since our f-stat is larger than this, of course we will reject. When do we reject? Remember, this is a one-tailed test, only one number to worry about. We are either above or below 2.76, and we reject if we are above. So because the f-stat is greater, then we can reject, and of course it's because we have sufficient evidence. Okay, so the next type of questions that you are very likely to get is this one, where you have a table that is only partially filled out. You get an SSW and you get an MSA. You might think, how do I answer that? Well, there's one more element of information you get from the text, and those are the number of groups, in this case 3, and the number of values in each group, and that is 5. And so you see these numbers, what do you think in your head? First of all, you can fill in these degrees of freedom. What is n? N is just 5 times 3, so that's 15. So n minus 1 is 14. N minus c is 15 minus the number of groups, so 15 minus 3, that's 12. And c minus 1 is going to be number of groups minus 1, that's 2. And now there are two ways of answering this. Either you go ahead and compute SSA as MSA times 2, then MSW, and SST as the sum of these two, and then the FStat. You can do this using your calculator, or you can be quicker and cleverer and just do it in Excel using this little template that we had. So 2, 12, 14, 156, 65. Okay, let's fill those in. 2, 12, 14. We didn't know this one. This one is just going to be the sum. Here, what did they tell us? 156. 156. Okay. They told us this one that was 65. So right here, just filling in these numbers, we found almost everything. We just need to add the SS here, which is this, times that, and we have all of our numbers, and our f stat is equal to 5. Let's fill those back in. 130, 286, 13, and 5. And voila, question finished. Last question on one-way ANOVA. This one actually asks you to compute an ANOVA in Excel. How do you know that? Because they gave you some data, so we're going to have to run the ANOVA. First things first, what is the hypothesis? Well, one thing we insisted on in the lecture is that the opposite of all the mu's are equal is not all the mu's are unequal. It's not all the mu's are equal. So this one is incorrect and this one is incorrect. It's going to be one of these. The only difference is, are there 4 mu's that we're comparing or 11 mu's that we are comparing? Well, look at the top. We have 4 categories. We have a main campus and 3 satellite locations. 4 locations. 11 is not the number of locations, it's the number of observations at each location. So definitely not this. Now everything else we're just going to get straight out of Excel. Let's click on our data and download it. Here I open my data. I'm going to do an ANOVA analysis. So I click on data, then I click on data analysis. Then I select ANOVA single factor, and that's one-way ANOVA. Click OK. Input range. I select all of this. Grouped by columns, yes. Labels in first row, yes. Alpha was 0.05. You can go back and check. And the output range, like I said, I prefer to add it inside the same spreadsheet. Press enter, and here it pops up. This is your one-way ANOVA. All of the answers are in this table. They want the f stat rounded to 2 decimals. There it is, 6.99. Be careful, this is the f stat, this is the f critical, the one to which the f stat gets compared. 699. The p-value, 3 decimals, 0.001. And of course, the p-value here is very low. It is a less than alpha. We reject because there is sufficient evidence. That's it. Okay, question 6. Now we are doing two-factor ANOVA. So these are two-way tables. Let us scroll down to our two-way ANOVA summary table. You recognize that this is similar to this. And again, this is a fill-in-the-blank kind of question. So let us go ahead and fill in the blanks. Here we actually have no text information. Everything you need to do are just relationships between these different numbers. So first things first, r minus 1 is equal to 4, and r minus 1 times c minus 1 is also equal to 4, which means that c minus 1 is equal to 1, because 4 is equal to 4 times 1. This clue unlocks everything else, because then we know that this plus this plus this plus that is equal to this number. And so this is going to be 34, 5, 9, 39. Again, my preference is to just go ahead and do it in Excel. So let's do that. Let me put in some row names. And let's go ahead and fill it in. 4, 1, 4, 30. Then what else do we know? We know SSB is 5, MSA is 10, MSAB is 45. And last thing, we know that this F stat is equal to 1. This F stat for the B factor. Again, this is going to unlock something because this cell is equal to whatever is here and whatever is here. And because this is 1, we know that these two are equal. So this is pretty helpful. What else can we fill in? This is equal to this cell times that cell. This we can compute the exact same way, so I'm just going to copy the formula. This is 5 divided by 1, so that's just 5. And because of this 1 here, we know that this is also 5, because 1 is equal to 5 divided by 5. And this unlocks everything else. This is equal to 10 divided by this, and this is equal to 45 divided by that same thing. Here, once more, we can just copy the form. And we've got all of our answers, so now I'm going to fill them in. Oh, I forgot. These sums of squares are also the sum, 375. And everything worked out nicely. This is by far the easiest way to solve these problems. Okay, question 7. Now this is going to get a little bit tough because we have 13 parts in these questions. But don't worry, it's actually fairly straightforward, and those questions always just rely on you running an ANOVA and then looking at the output of an ANOVA. So here you know it's a two-way ANOVA because they tell you about factor A, developer strength, factor B, developer time, which are impacting the density. That's your Y variable. So you have your three variables, the density, factor A, and factor B. It also tells you that there are 4 replicates in each of the 4 cells. Remember the cells being a combination of factor B and factor A. So let us open this data and run an ANOVA. First things first, what are the hypotheses? Well, the question is there an interaction between developer strength and developer time? Remember, the first question is always about the interaction. So since this is what we are trying to prove, this is going to be H1. H0 is going to be the opposite of that. So there is no interaction is our H0. And if we have enough data, if we have sufficient evidence, we reject that and prove that there is an interaction. So this is our C. And now we just have to run the ANOVA and answer the questions. Okay, so here I pulled it up in Excel so we can just follow. Let me run the ANOVA. This time it's an ANOVA with 2 factors and replication. What is my input range? Again, that's my whole data. What are the rows per sample? That is the number of rows for each category of this factor, right? It's the same thing that they call the replicates. So there are 4. Alpha is 0.5, and the output range, once again, I prefer to put it somewhere on the spreadsheet. And here is my ANOVA. They want the f-stat with 2 decimal places. Now be careful, which f-stat do they want? This one, third one. We are talking about the interaction first, so it's the third line, the first line. So here's 1.50. What is the p-value? This? Let me just change the format to 3 decimal places here. They want 3 decimal places, 0.224. Nope, 0.244. 0.244 is a lot larger than 0.5, so this is not enough to reject. Therefore, it's going to be one of these two, which one do not reject because we have insufficient evidence to do so. So here's the most important thing. Since we could not reject the null, since we could not say that there is interaction, now we can look at these 2 lines, the effect of factor A, the effect of factor B. You're only able to do this because we failed to reject this hypothesis about the interaction. Okay, so let's do it. Is there an effect due to developer strength? Developer strength is our first factor. It's our first line. Is there an effect? Yes. Look at this p-value. So what is the hypothesis? Again, the hypothesis is that there is no effect versus not this. We're not saying it has to be negative. We're not saying it has to be positive. It's just no effect versus an effect. Value of the test statistic, 37. Oh, they want 2 decimals. P-value 0.000, they want 3 decimals. This is very low. We reject. We have sufficient evidence. Same thing for development time, but that's factor B. This is this column. The p-value is too high. Clearly, no effect. What is the hypothesis? It's no effect versus an effect. Here. F-stat 0.3 8, rounding up to 2 decimals here. P-value is 0.552. And of course, we cannot reject. Okay, so here comes the last part of the question where they ask us to figure out which of these plots is the correct one. Now you can do it the direct way where you actually compute the numbers in the 4 cells and see which picture corresponds. So let's do that. So I prepared a mini table here. This is the average. I drag it out to get the 14.
This is the average. I drag it around to get this one.
So for strength equals 1, I have 3.5 and 3. And for strength equals 2, I have 7.5 and 9. So we have to have a full line that goes from 3.5 to 7 and a dotted line that goes from 3 to 9. Our scale is 0 to 10. 3 is around here. So these lines are not starting at 3. These lines are not both starting at 3. These lines, it seems the blue line is a little too low. This one actually seems to work. Blue line going from 3.5 to 7.5 and a dotted line going from 3 to 9. This makes sense to me. Note, however, that you already could have had a clue about this. Why? Because you know that sample has a strong impact. Sample is what? The first factor, so developer strength should have a strong impact. Therefore, you know that it cannot be this one because you don't see a clear rising pattern between strength 1 and 2. Here, you don't see a clear rising pattern between strength 1 and 2. So you already knew from the get-go that it had to be either this one or this one because they're the only ones who show a strong increasing impact of strength. There's a last small part related to this. Developer's strength has a significant positive effect. The development has no significant effect, and the difference between the densities development of 14 and 10 minutes using a developer strength 2 is not significantly different. What are they trying to say here? They're trying to ask whether there's an interaction effect. There isn't. Okay, I'm not going to go over 8 in detail because it's actually almost exactly the same. Here is your data. Here is your two-way ANOVA. These are all the answers. The only difference is that in this case, it's a little bit harder to figure out which plot is the correct one. That's it. Hopefully this is helpful.